Good day, everyone. Now that we have discussed the basic structures of bacteria, you know already how bacteria are classified into gram-positive, gram-negative according to gram-staining reactions. You are now ready to learn more about the different bacteria, the specific bacteria no, under gram-positive and gram-negative. So for today, I will be discussing the basic test on how to identify gram-positive bacteria. Let's start with by recalling what gram-positive bacteria is. So remember, we learned that gram-staining reaction would actually render a particular bacteria as either gram-positive or negative, depending on the content, no, of the amount content of that specific membrane structure we call peptidoglycan layer, such that upon addition of your primary stain, so this is just a review, upon addition of your primary stain, since your gram-positive bacteria has a thicker peptidoglycan layer, approximately about 40 sheets, and that 40 sheets of peptidoglycan layer comprises 50% of the total cellular volume, okay, cell membrane volume of a gram-positive bacteria. Chances are it's going to take up the stain, and upon addition of a mordant that crystal violet forms complexes with the mordant iodine forming crystal violet iodine complex. And since that complex now is actually a high molecular weight substance, being initially surrounded with or covered with a thick peptidoglycan layer, it could not actually go out from the cell membrane. You can still recall when we added a decolorizer, which is an organic solvent, no, particularly alcohol or acetone, chances are the proteins in the peptidoglycan layer will coagulate, will be denatured, and so making the membrane not not readily making the membrane more resistant to any leakage of substances inside the cell. And so the CVI complex will be retained inside. And so upon addition of a secondary stain, which is safranin, chances are it could not penetrate inside the cell. And so a gram-positive bacteria would retain the color of the primary dye, which is your crystal violet. And so after the entire gram staining procedure, a gram-positive microorganism will most likely color violet. Okay, gram-positive. Whereas versus gram-negative, it will take up the secondary stain because the outer membrane has been dissolved by the decolorizer, which is an organic solvent. And so your CVI complex now could escape the cellular membrane, and this would lead to a more leaky membrane such that safranin now could gain entrance to the membrane, rendering the bacteria now, if it is gram-negative, colored red. Okay, so I hope that's clear to you. Now, in general, this diagram would summarize the entire concepts, basic concepts on how we classify gram-positive microorganisms. So remember, gram-positive microorganisms may be cocci. No? All cocci are gram-positive except for Moraxella, Neisseria, and Veillonella. Remember? And then a bacilli could also be gram-positive. Again, if we recall the general gram staining rule, all bacilli should be gram-negative, but there are exceptions. The except Clostridium, okay, Bacillus, Listeria, Corynebacterium, Erythrospilothrix, Propionibacterium, among others, many. And then under gram-positive, we also have this what we call those bacteria producing branching filaments. No, we actually put them under different classification because they form filaments, they form branches like a normal fungi would do. Okay, so in general, a bacilli, a cocci, organisms with branching filaments may be gram-positive, no, depending on the color it retains after the gram-staining reaction. Now, under each sub-classification, we may classify these according to their ability to utilize oxygen, no, to survive, to grow in the presence or absence of oxygen. Again, when you say aerobic, these organisms actually are tolerant, okay? They are capable of surviving in the presence of oxygen. We say obligate aerobes would mean they live, no, they should live in the presence of oxygen. And absence of oxygen would mean these organisms would actually die. Okay? So that means they are dependent to the presence of oxygen for food and energy production. And when you say facultative aerobic, these organisms may actually utilize oxygen if present, no, for energy production, but when absent, they could still live without oxygen. No, they actually undergo fermentation for energy production in the absence of oxygen, meaning with or without oxygen, it could live. It has its ways of producing food and energy even with or without oxygen. And then when you say tolerant, no, aerotolerant, these organisms actually can survive, no, in the presence of oxygen. We say micro aerotolerant or micro aerophile, they only survive in the presence of small amounts of oxygen. I hope you could recall that. No? We've already discussed that in Unit 3. Now, when you say anaerobic, these organisms most likely do not survive well, could not tolerate well the presence of oxygen. So when you say obligate anaerobic, no, these organisms actually do not contain what enzymes? They do not contain superoxide dismutase, catalase, peroxidase, such that free oxide radicals, which are produced in the presence of oxygen, they could not tolerate it, and so they die. That's why they are called obligate and anaerobes, meaning anaerobes, they survive. No, obligate, they survive in the absence of oxygen. Even the lowest amount of oxygen is detrimental to these organisms. And when you say facultative, anaerobe facultative would mean they can tolerate, they can use oxygen uh, as an element, no, for energy production if present, but when absent, they could still survive, no, through fermentation. Okay, you say um, facultative again, they could survive with or without oxygen. Okay, now having said all of those things, now I want to direct your attention on, and then we have nocardia and actinomyces under branching filaments. And say acid fast, no, this is actually another type of staining which we will discuss in the next few chapters. But again, to differentiate the two, nocardia is aerobic, actinomyces anaerobic, weakly acid fast, meaning it takes up um, the primary dye in acid fast weakly, it's lightly red. Uh, However, actinomyces do not. It's not acid fast, no negative acid fasting risk. Now, I want to direct your attention on this area here, cocci. This is actually our discussion for the entire week, no, cocci. So cocci in general, remember, if you can still recall our basic discussion on bacterial cell morphology, they actually appear rod-shaped, no, upon gram staining. When you gram stain, then you look at the microscope, they appear rod-shaped. And on the basis of morphology, they could be further classified such that if a cocci now um, forms clusters, okay, we term it as staphylococcus. If they form chains, we term it now as streptococcus. Okay? So you learned also before that under cocci, we also have cocci in pairs, no? Diplococci. Very common organism is Neisseria, but again, that's gram-negative. That's why we're not including it. But you also learned before that we have gram-positive cocci in short chains, no? Could be short chains, no? Could be in pairs or longer than that, no? Particularly, very good example is your strep pneumoniae. And then we also have cocci in clusters of 8, no? Urosarcina, lutea. These are gram-positive cocci. And then we also have cocci in 4, in groups of 4 or clusters of 4. That is basically your Gaffia, no? Gaffia tetragena. Now, the main focus of this lecture is to actually discuss, no, the basic tests that we use to classify cocci as either Staphylococcus or Streptococcus. We will not actually discuss the organisms in this lecture, no? We'll have a separate discussion for these, no? It needs to be discussed face to face. But um, this lecture will actually give you an overview of the different tests that will help us identify if the organism belongs to either genera Staphylococcus or Streptococcus. Let's start with Catalase. Okay, so Catalase, if you can still recall, the word "ase" there denotes it's an enzyme. Okay, so Catalase, if you can still recall your basic biochemistry, is an enzyme that mediates breakdown of, again, catalase. It mediates breakdown of hydrogen into water and oxygen. In other words, any bacteria which contains catalase enzyme would produce a positive catalase reaction. Got that? And a positive result. Remember what's the byproduct of hydrogen peroxide? Um, Degradation by catalase, water, and oxygen. And so a positive result for catalase would mean bubble production. Now, if you have bacterial culture and the you added the reagent, which is hydrogen peroxide, you are most likely to infer that that organism has catalase enzyme, or the organism is catalase positive if it produces bubbles. The bubbles there would actually represent the byproduct of your hydrogen peroxide breakdown, which is oxygen, and the liquid portion water. Okay? So catalase positive organisms, if it is catalase positive, then you're most likely to think of Staphylococcus as the organism being isolated. But if it does not produce bubbles or a weak bubble production upon addition of hydrogen peroxide during catalase test, would mean that the organism is non-catalase producing, and so we most likely would think of Streptococcus genera. Right. Now, let's go to Staphylococci. Remember, the test that would differentiate Streptococcus from Staphylococcus is Catalase. Now, let's go to Staphylococcus. You've heard, no? You've read before that under Staphylococcus, we actually have a lot of species. Now, I will be discussing in the next lectures these medically important Staphylococcus organisms. But for now, I want to focus on the test that will classify these Staphylococcus as to different types. No, so we have another test we call coagulase. Okay, from the word itself, is it's an enzyme. Coagu means it causes coagulation of a certain protein. Therefore, coagulase is an enzyme which causes coagulation of proteins. And later in the next few slides, I will tell you what that protein is what protein is being coagulated upon addition of the reagent. And so we could say if that protein is coagulated, that would mean that the organism has coagulase. Okay? Now, in other words, if we say coagulase, if catalase positive reaction is denoted by production of bubbles, coagulase from the term itself, coagulate, the positive reaction is the presence of a coagulum. Okay? So, if the test sample, the test organism produces coagulum, that would mean upon addition of a reagent, that would mean that the organism has coagulase enzyme. And a coagulase enzyme would mean coagulase positive result. The organism producing a coagulase positive result okay, among Staphylococcus species is your Staphylococcus aureus. Okay? Now, other Staphylococcus species which do not produce the enzyme coagulase are termed as coagulase negative Staphylococcus or CONS, no? C-O-N-S. The C-O-N-S would mean coagulase negative Staphylococcus. Under coagulase negative Staphylococcus, we have Staphylococcus saprophyticus and Staphylococcus epidermidis. Now, there is a need to differentiate the two, and the test that could differentiate saprophyticus from epidermidis is the antibiotic novobiocin. Now, we say novobiocin sensitivity, we say sensitivity, the organism is inhibited, the growth of organism is inhibited, or the organism itself is killed, no? By the antibiotic novobiocin. So, when you say novobiocin sensitive or novobiocin sensitivity positive, that would mean that the organism is sensitive to the effects of novobiocin. And the best model organism that shows sensitivity to novobiocin, meaning it is not resistant to the effects of the antibiotic novobiocin, meaning it's killed by the novobiocin antibiotic. The growth is inhibited because it is sensitive to the novobiocin antibiotic. The model organism is your Staphylococcus epidermidis. But you say novobiocin sensitivity negative or novobiocin resistant, meaning the organism is resistant now to the effects of novobiocin, or to put it simply, the growth of the organism is not affected by the antibiotic novobiocin. It's not killed by novobiocin. That is now your saprophyticus, Staphylococcus saprophyticus. But in general, these two are coagulase negative Staphylococcus because they produce a negative reaction during coagulase test. Okay? But to differentiate the two, we use novobiocin. Now, let's go to the other side. Okay? Again, the test that would differentiate Streptococcus from Staphylococcus is Catalase test. And when you say Catalase positive, we are actually referring to the genera Staphylococci or Staphylococcus. Now, organisms producing Catalase negative reaction, okay, are classified under Streptococcus. Now, I want to limit the discussion between Catalase positive and negative as Streptococcus and Staphylococcus only. It's because we actually have a lot of organisms which produces catalase positive reaction, catalase negative reaction. But for this discussion, let's just limit on cocci, no? Cocci producing catalase negative and positive reactions. Okay? So I will show a list later of other organisms which do not belong to the genera Streptococcus and Staphylococcus, which produces also catalase positive or negative reactions. Now, we say catalase negative, the organism do not contain the enzyme catalase, and so upon addition of hydrogen peroxide, what's likely to happen is that there is no production of bubbles, or there is weak production of bubbles. That is particularly true to the genera Streptococcus. Okay, now we basically classify the different species of Streptococcus according to its ability to lyse a certain substance. So we call that test now hemolysis. Now, lysis or lysis would mean it's capable of lysing hemo red blood cell. It's capable of causing bursting or destruction of your red blood cells. Hemolysis, therefore, okay, would detect the presence of a substance that is capable of hemolysis present in the bacteria we call hemolysin. Hemolysin is the substance that is responsible for hemolysis in Streptococcus bacteria. Okay? So under hemolysis, we actually classify it according to the degree of hemolysis. So when you say alpha hemolysis or alpha hemolytic organism, meaning that organism is capable of partial hemolysis of your red blood cells. Okay? Red blood cells, because the culture media that we use to demonstrate hemolysis is blood agar. Yes, correct. No, you heard it right, blood agar. It's actually in an agar wherein we added blood. And the most preferred type of blood is actually a sheep's blood. Now, since we're um, a developing country, okay, it's really expensive to use sheep's blood every time we demonstrate hemolysis. So we could actually use human blood, no, because it contains red blood cells to demonstrate hemolysis. But the most preferred is actually your sheep's blood. Now, if the organism is not capable of fully lysing the entire red cells in the agar, we call it, no, it produces partial hemolysis, and we call that alpha hemolysis, no, partial hemolysis only. And the color actually is green, meaning when you say lysis, remember there should be clear, no, clearing of the clearing of the area of the agar we're in, surrounding area of the agar we're in, the organism is inoculated. Okay, so if it's colored green, that means there is only partial hemolysis. So that's alpha hemolysis. We say beta hemolysis, meaning the all red cells are surrounding the colonies are completely lysed, and so since red blood cells are completely lysed, there is clearing in the surrounding area. And if the organism is not capable of hemolysis, meaning if it's not capable of hemolysis, it does not contain hemolysin, and so we term it as gamma hemolysis, gamma absence of hemolysis. Now under hemolysis, alpha hemolytic streptococci, okay, we have this what we call Streptococcus pneumoniae and peridans, Streptococcus mutans and Streptococcus mitis. Now, the tests that will differentiate these organisms are the following. We have optokin and bile solubility. Optokin is actually an antibiotic. No? So, should always bear in mind that if a word is followed by the term sensitivity, we're actually talking about antibiotics. So, optokin sensitivity, when we say the organism is optokin sensitive, that would mean that optokin now is able to inhibit growth of that microorganism in the culture media. Okay, meaning it is sensitive to the effects of your optokin. And that is usually seen as clearing of the area surrounding optokin. No? And we say bile solubility, addition of bile, no? The reagent here is bile. Addition of bile would cause solubilization. The organism becomes lysed, no? Becomes insoluble to bile. And so what you're most likely to see in a test tube upon addition of bile. If it is soluble to bile, you see clearing. Okay? But if it's not soluble to bile, you'll see turbidity. Turbidity would mean that the organism was not lysed with bile. So an organism, an alpha hemolytic streptococcal organism that is sensitive to the effects of optokine and soluble to bile is your Staphylococcus, Streptococcus pneumoniae. Okay? And resistant to the effects of your optokine and insoluble to bile are your very dense group, S mutans, Streptococcus mutans, and Streptococcus mitis. Under beta hemolytic strepto, B, beta for bacitracin, we use another antibiotic we call bacitracin, such that the organism which are sensitive to the effects of bacitracin, meaning if it is sensitive to bacitracin, the organism growth is most likely inhibited by bacitracin. And inhibition on the plate, the culture media, is likely noted by a clearing of the surrounding area, the area surrounding the bacitracin disc. And the organism, according to beta hemolytic streptococcal organism, according to bacitracin sensitivity, we could classify it as group A or group B. So the organism sensitive to bacitracin, okay, is your Streptococcus pyogenes, also termed as group A Streptococcus, group A beta hemolytic streptococci. Okay, those are the other names of strep pyogenes. And then resistant to the effects of your bacitracin is your group B beta hemolytic streptococci or group B streptococci or Streptococcus agalactiae. Okay? Remember those terms. You have to understand, that these are actually foreign to you. That's why I'm not actually rushing things, and I really wanted to introduce things to you slowly so that you'll get to appreciate more the subject. Okay? So next we have gamma hemolysis. Again, gamma would mean that the organism does not produce hemolysis on blood agar. And so if it does not produce hemolysis in blood agar, it does not contain the substance hemolysin. So under gamma hemolysis, we have another sets of microorganisms. We have your Enterococcus and non-Enterococcus. They are actually classified according to their growth in 6.5 sodium chloride. Now, if 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 the gamma hemolytic microorganism produces turbidity or growth in 6.5% NaCl, meaning it's positive for growth, it's classified as group D, or Enterococcus. And under group D, we have Enterococcus faecium, Enterococcus faecalis. Now, if it is sensitive to the effects of NaCl or sodium chloride, meaning the organism is killed, no? The organism is killed upon exposure to 6.5% NaCl, then it produces negative growth. Okay? So we classify it as non-Enterococcus gamma hemolytic streptococci. An example organism is your Streptococcus bovis. So, as early as now, before Thursday, okay, we will have a quiz on Thursday. Take note of that. You have to memorize the entire table, here, diagram here. Memorize this by heart. Okay? Now, let's discuss these basic tests that we use to classify an organism as Streptococcus or Staphylococcus, particularly your Catalase, Coagulase, and Hemolysis test. Let's start with your Catalase test. So again, as I've mentioned, Catalase test demonstrates the presence of the enzyme Catalase. So again, the enzyme is capable of breakdown of hydrogen peroxide into oxygen and water. So remember, in in a microorganism, once exposed to a superoxide radical, the superoxide radical is broken down into hydrogen peroxide by the enzyme superoxide dismutase. And the hydrogen peroxide in turn, okay, is broken down into water and oxygen by peroxidase and catalase. Okay? And again, if you can still recall, which particular organelle do we see these enzymes? Correct, no? In peroxisomes. So, as I've mentioned before, catalase production is actually a protective mechanism of bacteria because remember, once a bacteria is exposed to oxygen, it produces free radicals, and these free radicals eventually are converted to hydrogen peroxide. Both, no, free radicals and hydrogen peroxide are actually detrimental to the cells, the bacterial cell. So we have two methods. Okay? Two methods for catalase test. No, we have tube method and the slide method. So in tube method, the reagent used here is hydrogen peroxide. So we look at here. Okay, so we place the organism in the stick, no, or a glass rod. We place the colonies in here, and then we dip it into, we submerge it into a 1 to 2 ml of hydrogen peroxide solution, and production of immediate, no, bubbles, no, production of bubbles would mean that the organism has catalase enzyme. Then we have slide method, okay? So in slide method, we use a wooden stick, okay? We do not use wire loop because the wire would actually produce false positive reaction. Why false positive reaction? Remember, your wire loop contains iron. And when iron is added with hydrogen peroxide, what's likely to happen is that it undergoes oxidation, and so it produces bubbles. Thus, the false positive reaction. And so we use in slide method now a wooden stick. Okay? So the organism is placed in a glass slide, and then we introduce the organism in the glass slide using a wooden stick, and then we place a drop, no, 1 drop of 3% hydrogen peroxide into the glass slide, and so if it contains now catalase enzyme, it will produce bubbles. Okay, so the term here is copious bubbles produced, meaning active bubbling, because any weak reaction, small amounts of bubbles are considered negative for catalase. Take note of that. Weak bubble production. Very few bubble produced. No, negative for that. So these are the organisms which are catalase positive. Remember, I mentioned that there are also gram-negative microorganisms or organisms which are non-staphylococcal and non-streptococcal, which produces catalase positive results. So these are these organisms. These are those organisms. Okay, so you need to memorize these organisms producing a positive catalase reaction. But again, very distinct, no? Also are those organisms which do not contain the catalase enzyme, your streptococcus and your enterococcus. So these are the things to consider when doing the catalase test. So remember, the organism should not be taken from blood agar. Why? Remember, your blood agar contains red blood cells. And you learned before that red blood cells contain also catalase. And so when you accidentally scrape off, no, in any portion of the agar, most chances are you also included red blood cells in the sample. And so since red blood cells contain catalase also, it will produce false positive reaction. Got that? So the culture should be 18 to 24 hours old. Why 18 to 24 hours old? Old culture should not be used because most likely these organisms um, these organisms now will actually produce a negative, no false negative catalase reactions. Hydrogen peroxide must be fresh because it is very unstable. What happens if you use an expired reagent? Chances are it will produce false negative result, no? Because it's not capable anymore of breaking down into oxygen and water. So I mentioned this already, no, that iron wire loop should not be used. It should not be used. Why? It's because iron oxidizes in the presence of hydrogen peroxide, and so it upon oxidation it produces bubbles. So false positive result. We don't want that. Some bacteria produce a peroxidase that catalyzes a breakdown of peroxide, causing reaction to be weakly positive. So again, I've mentioned that a weakly positive reaction, no, meaning there's only a few bubbles produced, is still considered negative. What's truly positive is copious production, active production of bubbles. So you do not add organism to the reagent, particularly if iron-containing inoculating loops are used, because this will cause false positive reaction. Okay? So I hope you're getting that. Then we proceed to another test we call coagulase. Remember again, if you can still recall in the second slide, that coagulase test will actually differentiate coagulase positive Staphylococcus, which is Staphylococcus aureus. No, it will differentiate Staphylococcus aureus from cons or coagulase negative Staphylococcus. That is because now of the presence of which enzyme? Coagulase, no? And coagulase, by the term coagulase, the positive result is presence of coagulum. Now, the mechanism of coagulase test, therefore, if it produces coagulum, is based on agglutination reaction. So meaning agglutination would actually mean formation of coagulum. So coagulase, okay, being an enzyme, is actually found, no, found in the surface of bacteria. And this is capable, okay, this is capable of converting fibrinogen. Fibrinogen is actually a clotting factor. Once it acts upon fibrinogen, fibrinogen being soluble, once acted upon coagulase now, okay, it produces now an insoluble polymer we call fibrin. Okay? So, to be specific, coagulase acts upon prothrombin, producing a complex we call staphylothrombin. This staphylothrombin activates protease. A protease cleaves fibrinogen, okay, through the process of proteolysis. Then fibrinogen is eventually converted into a clot, a fibrin clot, which is insoluble. This is now the product that is visible to the naked eye. So, as to the types, we actually have two types. We have this what we call the bound coagulase and the free coagulase. Now, coagulase positive organisms, so either bound or free, they actually produce a positive coagulase test. Now, how do we differentiate the two? You say bound coagulase, it refers to a clumping factor whose presence becomes evident after performing the slide coagulase method. Okay? So, we say bound coagulase, we actually refer to a certain clumping factor. And to detect this clumping factor, we perform the slide coagulase method. Now, another Another pertinent information that you should take note of, we'll discuss more of these in when we discuss Staphylococcus, okay? Another pertinent characteristic of a bound coagulase is that it's stable, no? It's stable against heat, and this is actually a non-virulent factor. If it's non-virulent, that would mean this bound coagulase, once present in our circulation in humans, this does not elicit a disease process, no, or an immune response. Okay, since it's non-virulent. Um, Being detected, okay, by a slide coagulase method, detection of your bound coagulase is actually a screening test to detect the presence of coagulase in a particular organism. Okay? Screening test. Then we have your free coagulase. Free differs from bound because when you say bound, this is actually attached to the membrane, okay? The membrane of the organism. So when you say free, this is present. This refers to the extracellular coagulase, okay? Meaning free, it's not bound, no? Extracellular coagulase, and the presence of your free coagulase is detected using the tube coagulase method. So take note na, bound coagulase, we use slide coagulase method. Free coagulase, we use tube coagulase method. No, we use a test tube. In bound, we use a glass slide. So a characteristic of your free coagulase is that it is unstable to heat and it is a virulent factor, meaning this is now the particular coagulase that triggers immune response causing a disease process in humans. So again, bound, clumping factor, slide coagulase method, stable against heat, and non-virulent. We say free coagulase, extracellular because it's free, it's not bound, detected using tube coagulase, it's unstable to heat, and it acts as a virulent factor. Okay. So slide coagulase versus tube coagulase. Again, slide coagulase is used as a screening method of the presence of coagulase in an organism, and this detects what type of coagulase? Bound. Tube detects which type of coagulase? Free coagulase. So again, slide coagulase detects cell bound. I'll keep on repeating that because we will have a thorough discussion on these enzymes, no, when we discuss Staphylococcus next meeting. Slide coagulase again detects cell-bound coagulase, which directly agglutinates the plasma into fibrin clots. So what we do in slide coagulase method is that a bacterial suspension is placed in a gas slide here. So this is now the gas slide. We place the bacterial suspension in here, and then we add a drop of plasma. So plasma is our reagent. Okay? And then any coagulation, any coagulum form, this is now the coagulum, is considered positive for the reaction. So again, this is just a screening method. No? This is just a screening test which detects presence or absence of coagulase. To look at the positive result, presence of clumps, no, or coagulum, absence of clumps in negative results. Then we have tube coagulase. So again, it detects the presence of free coagulase. Free coagulase is also called staphylocoagulase, okay, which indirectly reacts with fibrinogen. Indirectly reacts with fibrinogen. Remember, in slide coagulase, bound coagulase directly reacts with fibrinogen, whereas your free coagulase indirectly reacts fibrinogen. So what we do here is this, okay? So we add the organism suspension into the test tube. We use a tube, that's why the term is tube coagulase. We add it in a tube, and then we suspend it with plasma. The plasma now contains your clotting factors, fibrinogen, which is converted to fibrin if there is presence of coagulase, and then we incubate it within 4 hours. So this test now is confirmatory to the presence of coagulase. Confirmatory, no? To look at a positive result, presence of clump, no, a clot, versus the negative reaction, absence of clot. So slide coagulase, again, screening test, tube coagulase, um, a confirmatory test for the presence of coagulase. Then we have hemolysis, okay? So again, this detects the presence of the substance hemolysin, which is capable of breakdown of red blood cells, okay? And this can be further classified according to the degree, to the amount of hemolysis present in the blood agar. Again, if it's partially, okay, hemolyzed producing a green color in the surrounding colonies, we term it as alpha hemolysis. And you know already what are the organisms producing alpha hemolysis. Again, we have your viridans, Streptomyces, Strep mutans, and your Strep pneumoniae. Then we have those organisms producing clearing of the surrounding agar, okay, no green, no, but clearing of the surrounding agar. We term it as beta hemolysis. The organisms producing beta hemolysis in blood agar. So we have they are differentiated through the use of bacitracin. Okay. So we have your group A, beta hemolytic strep or strep pyogenes, or group B, beta hemolytic strep or strep agalactiae. Then we have those organisms which do not. So you see, there's actually no changes in the color of the agar. Okay, that means they are not capable of breakdown of red blood cells. And so if they are not capable of breakdown of red blood cells, they do not contain hemolysin. We term it as gamma hemolysis. And under gamma hemolysis, we have your enterococcus. So beta hemolysis, I've mentioned already, no, is usually associated with complete lysis, and we detect actually two hemolysins here, the hemolysin O and hemolysin S. No, hemolysin O is inactive in the presence of oxygen, and so we usually detect this through stabbing of the plate or agar. So you learn more of this, no, when we discuss Streptococcus. So examples of organisms which produces which are beta hemolytics. I've mentioned this already. No? So your group A beta hemolytic strep or your gas, and then Streptococcus pyogenes. And then weakly beta hemolytic, we have Streptococcus agalactiae. We also have another from another genera, no, Clostridium perfringens, Listeria monocytogenes. So aside from uh, these reactions, no, I've made mention also of the different laboratory tests that will differentiate the different streptococcus, no, not just through hemolysis. So we have bacitracin, octokin, sodium chloride, okay, and bile solubility. Okay, so that's all for today's lecture. I hope you you've appreciated these basic tests, no? And as an exercise, this table summarizes what will happen during alpha, beta, and gamma hemolysis. So I've mentioned this already. Okay, so keyword for alpha is incomplete or partial, green color. Keyword for beta is complete, since red blood cells completely lyse, there's clearing or colorless zone around the colony. Gamma, no hemolysis or no change in color. Okay, so as a summary, to sum up everything, let's answer these exercises. No, future examinations will be like this, okay? Because we need to apply what we have learned. So first question, a 28-year-old male with a history of chronic alcoholism presents with fever and productive cough. He was apparently well under 3 days prior to consult when he developed cough, shaking, chills, and headache. His cough has been associated with the production of yellowish, greenish sputum, which occasionally was blood tinged with brownish streaks. Blood culture grew gram-positive cocci in pairs and short chains, which laboratory tests will facilitate identification of the genes. Now, if you are presented with examination questions, which are cases, no, do not fret. Do not get intimidated. I told you already, no, I've taught you how to approach these types of questions. You just have to select the keywords. So the keywords here is that the organism and that the patient has a fever and has a cough. And these are the symptoms, no? With yellowish green sputum. So most likely, the patient has obo with phlegm. Most likely, we're considering pneumonia, no? Pneumonia. But again, at your level, you could not diagnose this patient. You just have to point out which are pertinent to the case. Now, this is now the very pertinent information here. Upon isolation, no, in blood culture, gram-positive cocci in pairs and short chains were identified. So the keyword here is gram-positive cocci, okay, in pairs and short chains. Now, having noted that these organisms are cocci, gram-positive cocci, the question is, which of these tests will facilitate identification of the genus? We need a test that will differentiate these gram-positive cocci. And for all we know, we only have one test which will directly differentiate cocci as to what are again the two cocci that we just discussed, Staphylococcus and Streptococcus. And what is that test? Is it coagulase? No. Coagulase test is used to differentiate Staphylococcus aureus from coagulase negative Staphylococci. Is it novobiocin? Your novobiocin test is actually a test that will differentiate coagulase negative Staphylococci, okay, into Saprophyticus and Epidermidis. So it's not actually gonna differentiate the cocci from strep and staph. Then we have bacitracin. Bacitracin is actually a test which will differentiate beta hemolytic, okay, streptococci. So correct answer here is Catalase test. No? The Catalase test will differentiate Staphylococcus from Streptococcus such that Catalase positive is your Staphylococcus, Catalase negative Streptococcus. Okay. Another question. A 32-year-old male presents to the emergency room with a high fever, chills, and severe pain and swelling in his right knee. He reports a recent injury to the knee while playing soccer. Examination, the knee is warm, erythematous, and tender to touch. Aspiration of the knee joints reveal purulent fluid. Gram stain of the fluid shows gram-positive cocci in clusters, and then further lab tests produce catalase positive, coagulase negative result. So which organism is most likely the causative agent of the knee infection? So again, it's a gram-positive cocci in clusters. So we're actually thinking of, let's just say gram-positive cocci. Let's break down terms. No, gram-positive cocci, we're thinking of either strep or staph. But it's in clusters, most likely it's Staphylococcus. Then the supporting tests are the following: catalase positive. So most likely it's Staphylococcus, no? Because Streptococcus is catalase negative. Then coagulase positive. You know already what I'm thinking. If it's coagulase positive. So let's break down. Do not answer yet, no? So let's rationalize everything. So we have Streptococcus pneumoniae. Catalase, is it catalase positive, catalase negative? Catalase negative. So this should not be the answer. Then we have Streptococcus pyogenes, still catalase negative, no? It's not catalase positive. Then we have Staphylococcus epidermidis. Yes, correct. It's catalase positive. So we're actually left with two answers, Staphylococcus aureus, which are both catalase positive, and Staphylococcus epidermidis. Okay, both are catalase positive. How to differentiate the two? Remember, we have this what we call coagulase negative Staphylococcus aureus. And a coagulase negative Staphylococcus aureus is true to your cons, which includes Staphylococcus epidermidis and Staphylococcus saprophyticus. So the correct answer here is Staphylococcus aureus. Oh, very easy, no? Sige, last. A 45-year-old female with a history of diabetes mellitus presents to her primary care physician with complaints of a painful erythematous lesion on her left foot. She reports that the lesion started as a small blister but has since become larger and more painful. On examination, there is a well-defined area of erythema with central necrosis and surrounding edema. Culture of the lesion yields gram-positive cocci in chains, catalase negative, showed beta hemolysis and blood agar bacitracin sensitive. Which of these organisms is most likely the agent on the foot lesion? So the patient has a foot lesion and we have isolated these organisms showing gram-positive cocci. So let's see if they are all gram-positive. Strep, staph, they are all gram-positive. Cocci in chains, so most likely we are thinking of Streptococcus, no? So wala na ning Staphylococcus here. Supported by the test catalase negative. Since your strep are catalase negative, we're most likely thinking of the genera Streptococcus because Staphylococcus is catalase positive. So, X na siya, no? We omit this. Then, it showed beta hemolysis. Uy, beta hemolysis. We actually have two organisms showing beta hemolysis on blood agar, no? The beta hemolytic strep. And these beta hemolytic strep are classified as group A and group B. Okay? And then, furthermore, this group A and group B are classified according to their sensitivity to bacitracin. So which is sensitive to the effects of bacitracin? Group A. Okay? Which are non-sensitive, meaning resistant to the effects of bacitracin? Group B. So, which of these organisms is the most likely causative agent? So, Strep pneumoniae is gram-positive cocci in chain. Yes, correct. But, yeah, it's also catalase negative, but it's beta hemolytic or not? It's alpha hemolytic, no? So, and then we have Streptococcus epidermidis, Staphylococcus epidermidis, staph, it should be catalase positive, okay? So it's not the correct answer. Although Staphylococcus aureus, okay, shows beta hemolysis and blood agar, but it's catalase negative, okay? Staph aureus also shows beta hemolysis and blood agar. So the most likely correct answer here is Streptococcus pyogenes because it's catalase negative, it's beta hemolytic and sensitive to the effects of bacitracin. But if the question says bacitracin resistant, no, your answer should be Streptococcus agalactiae or group B GBS, no? Group B beta hemolytic strep. That ends our session for today. Thank you very much. If you have questions, you just message me. Thank you and God bless.